We'll finish up in lab on Thursday. In lab this week, we're going to be doing nothing but looking at tissues under microscopes so that we can find these tissues and learn these tissues. I can tell you that this portion of the material from a lab test perspective, I'd be surprised if there's anything less than 15 questions on the lab exam. When you think there's 52 questions on a lab exam, 15 of them are dedicated to tissues. That's a huge percentage. That's 30 points on your first lab exam. So what we're teaching today in lab lecture, what we're doing in lab this week plays a huge role in your first lab exam, which I believe is a week from Thursday, so 2 weeks from today. The first lab exam is, I think, 2 weeks from today. It's definitely not yet, it is, because it's not next Thursday. Regardless, well, no, it's not next Thursday. Whatever it is, it's coming. I'll let you know when we get to lab. Anyhow, so on the lecture exam, yes, I'm going to ask questions about this. The primary question I'm going to ask on lecture from this part of the chapter is going to come from function. I'm a huge function guy. So as I introduce a tissue, I'm going to tell you, well, this tissue is great because it has this function. The other thing that I'm a really big fan of with regards to lecture exam questions from this stuff is, where do you find these tissues? So I'm gonna introduce each tissue, and I'm gonna say, hey, look, this is a tissue that you find in the small intestine. This is a tissue that you find in the trachea. This is the tissue that you find in the bladder. We're gonna talk about where we find these tissues. That's the main thing I'm gonna be concerning myself with in lecture. Lab exam, it's primarily consisting of can you identify it under a microscope? So as we take a look at all these tissues, can you identify them under a microscope? And can you tell me where they're located? Those are the big two questions that they like to ask. The third one, which they also do ask, is which category of tissue does each of these individual tissues belong to? And here are your 4 categories of tissues. When we think about our tissues, they fall into one of 4 categories of either epithelial tissues or epithelium, connective tissue, which is a kind of a catch-all, muscle tissue, there's 3 varieties of muscle we're going to talk about, and nervous tissue. There's really only one nervous tissue, and that's the neuron. And every now and again, what they'll do in lab is they'll show you a tissue. Typically for each tissue that they question you about, they ask you 2 questions per tissue. The first question is typically, identify this tissue. The second question they usually ask on the same tissue is, where do you find it? Or what category of tissue does it belong to? If they ask you for the category, one of these 4 is what they're looking for. The first category of tissue is called epithelial tissue. And epithelial tissue is tissue that in general lines some part of the body. Either it covers the body like our skin, it lines the inside of the body. Think about the inside of the mouth, think about the inside of the digestive system, the inside of our respiratory system. It lines the inside of some part of the body, or it makes up the inner lining of our glands, whether it's a salivary gland. These are going to be the cells that are on the very, very inside of our salivary glands that are secreting the saliva, or these are going to be the tissues that are on the inside of our pituitary gland with our endocrine system that are secreting the hormones that pituitary gland releases. And when you think about their functions come from everything from protection. Epithelial tissue, think about our skin. Our skin's a phenomenal protectant. We're absorbing things. Think about our small intestine, which does a lot of absorbing of carbohydrates and fats and proteins and minerals and vitamins. We're filtering. Think about our kidneys. Or we're secreting something. Think about a salivary gland, which secretes saliva, or an endocrine gland that's secreting hormones. And so each of these tissues, as we go along, we'll kind of talk about, hey, this tissue is really good at doing this tissue is really doing that. But ultimately, these tissues as a whole are going to be doing one of these 4 functions or multiple of these 4 functions. And so when we think about a little bit deeper into these, they're going to cover or line our bodily surfaces. They're going to form sheets of cells that are linked together. We're going to have cells that are linked together forming sheets of cells. They tend to be avascular, which means they don't have direct blood supply. And these are cells that usually regenerate. Well, they all regenerate very, very easily, provided they are given the nourishment that they need. So these are cells that can get destroyed, but then can generally regenerate very, very simply to kind of make themselves whole again. Unlike some other tissues, like nervous tissue, which doesn't regenerate very easily, even if it's well nourished. And when we think about naming our epithelial tissues, there's a couple key things we need to think about. The first thing we need to think about is the number of layers. Well, let's go to the other one first. So the first one we're going to talk about is the shape of the cells. So when naming epithelium, epithelium is named by two key ingredients. One is the amount of cells or amount of layers of cells. The other one is the shape of the cells. And there's really three very, very simple shapes. Shape number one, we're going to call this a squamous cell. And squamous ultimately means flat. So here we have a basement membrane. What we're going to see are a series of cells that are flat. They are wider than they are tall. And we call these a squamous cell. The second type is called cuboidal. And cuboidal, as their name suggests, these are cube-shaped. Now we're looking at cells that are as tall as they are wide. They're shaped like a dice. And the last of our shapes is called the columnar cell. And as their name implies, these are cells that are shaped like columns. So these are cells that are going to be taller than they are wide. And so when naming epithelial tissue, the one thing you need to look at is what's the shape of the cell that you're dealing with. If you're dealing with a flat cell, we're dealing with a squamous epithelial tissue. If we're dealing with a square-shaped cell, we're dealing with a cuboidal epithelial tissue. And if we're dealing with cells that are taller than they are wide, we're dealing with a columnar epithelium. The second big thing that comes into play is the number of layers. If you have a single layer of cells, i.e., here's the basement membrane, and we have one layer of cells, it is referred to as a simple epithelium. On the same token, if we were to start stacking these tissues, and now we have multiple layers, we refer to this as stratified. So, if we were to have a single layer of flat cells, that tissue would be called simple squamous epithelium. On the same token, if I have flat cells and we have multiple layers, we would call it stratified squamous epithelium. A single layer of cube-shaped cells is going to be called simple cuboidal. A single layer of column-shaped cells will be simple cuboidal. And by and large, it makes these tissues rather, very, very easy to name. So again, when you're looking at a cell, or you're looking at a tissue rather, the first thing you want to ask yourself is, are we dealing with a single layer? Like we see up here, this would be simple. This would be an example of what tissue? So it's squamous because they're flat. So what would the tissue be called? Simple squamous. It's really that simple. On the same token, as we look down here, multiple layers. So this is going to be called stratified epithelium. This would happen to be simple, this would be stratified squamous epithelium is what we're looking at down here. And so as we name these tissues, especially the first 4 or 5 of them, this is going to definitely be our guiding principle. The second one, of course, is the shape of the cell. If the cell is a flat cell, we call it a squamous cell. If it's a square-shaped cell, we call it a cuboidal cell. And if it's a tall, thin cell, we refer to it as a columnar cell. So naming epithelial tissue is not that difficult. The key here is going to be, can you recognize each of these shapes as you look at them under the microscope? Some people have an easier time than others. It really just takes a little bit of repetition. The good news is we have, I don't know, probably 15 or 17 different tissues we're going to look at. 6 of them are epithelial tissues. So there's going to be some epithelial tissues on your next test. This just gives you a simple little box here which shows you the varieties that we're going to talk about here in lecture and lab. Gives you an idea of where they are and what they kind of do for us. But we're going to take them one at a time. So, the first tissue is simple squamous epithelium. So simple means what?
One layer.
One layer. Squamous means what?
Flat.
Right, so we have a single layer of flat cells. So when we think about simple squamous, heads up.
Sorry.
You're fine. So when we think about simple squamous, we're talking about a single layer of flat cells. And the one thing squamous epithelium is superior at doing is diffusion. The one thing that simple squamous does better than any other tissue is it is fantastic at diffusion. So any place in the body where we need to do diffusion, we're going to find simple squamous epithelium. And what is diffusion? We talked about it last week.
Like separate or
Moving one thing to the over a period.
You're getting closer. All right, so we're moving what from one place to the next?
Solute.
We're moving solutes. We're moving, think about a solution, right, where we've got a higher concentration on one side and a lower concentration on the other side. Remember how we talked about, I kept using this. Here's A, here's B, a lot of particles over here, just a handful over there. We're going to move particles across the membrane. This membrane is going to be made of simple squamous epithelium. If we're diffusing, if we're using diffusion in the body, you can bet that the material that we're using to make this is going to be made out of simple squamous epithelium. So simple squamous epithelium is going to be found in areas where we do diffusion. And there's a handful of really, really good places where we do this. First off, our capillaries. What is a capillary? It's a type of what? You've heard of the term capillaries before, have you not?
yes.
So what's a capillary?
Blood vessel.
It's a type of blood vessel, right? It happens to be the smallest type of blood vessel in the body. So as an artery, which is a big blood vessel, delivers blood to a tissue, it turns into a capillary. And a capillary is made up of nothing but simple squamous epithelium. And why do we deliver blood to a tissue? What's the point of bringing blood to a tissue?
So it can function.
Oxygen.
So we can bring oxygen to it. Well, how does oxygen get from the blood to the tissue?
Diffusion.
Diffusion. Oxygen will literally diffuse out of the blood into the tissues. So will the nutrients, like glucose and all those different things. They're going to diffuse out of the capillary and into the tissues. Meanwhile, carbon dioxide and waste material from the tissues will diffuse into the capillaries. So that's the way we ultimately can exchange waste material and nutrients, oxygen, and carbon dioxide at our actual tissues. The second great place where we find this is in the lungs. specifically something called an alveoli. What's the main function of our lungs? Why do we have lungs? To breathe. And why do we breathe? Why do we breathe? What's the function of breathing? It's got a very simple function.
Oxygen.
We need to bring oxygen into the body, and we need to get rid of carbon dioxide, do we not? So if I take a deep breath, and I bring all the oxygen in my environment into my lungs, why do I need to bring oxygen into my lungs? Where does that oxygen need to go? Into the blood. How does it get from my lungs to my blood? Via diffusion. And there's a special structure within our lungs called alveoli. And the alveoli are made up of nothing more than simple squamous epithelium, which allows for oxygen to diffuse from my lungs into my blood. And it also allows for the carbon dioxide in my blood to diffuse into my lungs, so we can exchange oxygen and carbon dioxide. Hence the reason why we breathe in and out in the base case. Bring in oxygen, get rid of carbon dioxide. Hands down, the best two examples of where we find simple squamous epithelium in the body are capillaries and the alveoli of our lungs.
How do you spell that?
Spell what?
Is alveoli?
A-L-V-E-O, A-L-V-E-O-L-I, alveoli.
Okay.
Yes, do these cells help to secrete and filter? Sure. But if you want to know the hallmark of this tissue's hallmark function, by far, is diffusion. And every place that we show an example of simple squamous epithelium, as we go through all the different organ systems, we're going to talk about them in areas where diffusion is taking place. So to kind of take a little closer look at it, again, what we're looking at is a single layer of tissues. Single layer, these cells are nice and flat. And what we're looking at here is actually an alveoli of the lung. An alveoli is simply a sac within our lungs. So this is an empty space, and it's surrounded by a single layer of flat cells. And this single layer of flat cells that's making this ring, and here's another ring, this is simple squamous epithelium. Surrounding this simple squamous epithelium, or surrounding this alveoli, made up of simple squamous epithelium, are blood vessels, specifically capillaries, both of which are made up of simple squamous epithelium, allowing for diffusion to take place. Oxygen moves from the lungs into the blood, carbon dioxide moves from the blood into the lungs, and that occurs via diffusion. And the reason why that is allowed to occur is because these tissues are made up of simple squamous epithelium, which is fantastic at allowing diffusion to take place. Everybody kosher with this tissue? Good. Tissue number 2. It's called simple cuboidal. So as we're thinking about this tissue, what is it going to look like?
It's gonna look like it has one layer in cubital.
Single layer of cube-shaped cells. So we're talking about a single layer. Cube-shaped cells. This tissue is spectacular at secretion and absorption. And it's going to be found in parts of the body where we have what are known as tubules. And what do you think a tubule is? It's a tube. But yules represents the concept that they're small. These are going to be microscopic tubes of the body. And we have these in a whole host of different areas. None so great as the kidneys. So when you're thinking about a place where you find them, you want to think about something called the kidney tubules. And in the kidneys tubules, the kidneys are made up of a series of hose-like structures that are referred to as tubules. And these tubules, think of them as a long hose. And imagine that hose, we cut that hose in half, and now we're looking down the barrel of that hose. The cells that make up that hose are a single layer of cube-shaped cells. We refer to this as simple cuboidal. And that's what we're looking at here. A single layer of cells that are cube in shape, simple cuboidal. And what we're looking at here are the tubules of the kidneys. We can see the basement membrane as it kind of makes its circle, and then the inner lining of that tubule is made up of a single layer of square-shaped cells. Here's another one, a third, a fourth, a fifth, a sixth, a seventh, an eighth, a ninth. And all of these tubes are, imagine them coming towards you, you're looking down the barrel of these tubes. Can everybody see simple cuboidal? These are a very unique tissue. When you see them, you really, once you see it, you can't unsee it. It's one of those things that once you see it under the microscope, it literally looks exactly like this. Some of these, when we get them under the microscope, there's some subtle differences than what we're going to see on the board. This one's not one of them. And what's great about these cells is these cells have the ability of absorbing and releasing material into the opening that's in the center. And the opening in the center is called the lumen. So the opening within this tubule is known as the lumen. And what we do in the kidneys, what's the function of the kidneys? To make what?
Urine.
You make urine, right? That's what the kidney's job is. And these tubules are important because they allow for us to properly make urine by adding to or removing from the fluid that's running through these tubules. Whatever's left is going to be urine that we get rid of. These cells have the ability of absorbing water, releasing stuff into these tubules so that we can make our urine and keep. Our body wants to keep the material that it wants to keep and get rid of the material it needs to get rid of. These cells do that for us. So as we're producing urine in the kidneys, the cells of these tubules will absorb and secrete material into the fluid of the kidneys, therefore producing urine.
What does that L word say?
Lumen, L-U-M-E-N. Lumen is ultimately the opening inside that tubule. Everybody kosher with simple cuboidal? Good. Which brings us to our third variety. Which is going to be simple columnar epithelium. So now we have a single layer of column-shaped cells. And these cells, there's a couple key things about these cells. First and foremost, they are phenomenal, again, at secretion and absorption. They just do it on a different scale. There's a couple of the things about these cells that are very, very important, especially when you're trying to identify them. First and foremost, this tissue is typically found along with something called a goblet cell. And a goblet cell secretes mucus. Any tissue that you look at, if you see a goblet cell, you're dealing with a columnar tissue. These tissues, also when you're trying to identify them, you are going to see cilia and or microvilli. And we talked about cilia and microvilli last week. What are cilia?
The little
Hairs.
The little hair-like structures. And what's the function of cilia?
to help stuff
Help
move.
move.
To move things that are on the outside of those cells. right? Back and forth. The example I used was our trachea, right? Well, when we look at trachea tissue today, you're gonna see the cilia on that trachea. What are microvilli? They're not hair-like structures, they're what-like structures?
Like
Finger-like structures. And what's the job of the microvilli? To increase a cell's surface area so that they better absorb. So when you're looking at these tissues, we're gonna look for, the one thing you're gonna see with these tissues, you're gonna see a single layer of very tall cells, and almost universally, these cells are gonna have goblet cells. You're gonna see goblet cells in all the tissues I'm about to show you. And you're gonna see either cilia or microvilli. And those two, if you can't see the cell in and of itself, that's one thing. But once you see cilia and microvilli, once you see the goblet cells, you know you are dealing with a columnar cell. Now it's just a matter of which one you're dealing with. With regards to places where you see simple columnar, the best example is our digestive system. Whether it's the stomach, whether it's the esophagus, whether it is the small intestine, large intestine, the inner linings of all of these organs of our GI or our digestive system are made up of a tissue that looks something like so. So what we see here is our basement membrane kind of running along the bottom with a series of tall cells, one layer of tall cells. Right here, these white cells, this is a goblet cell, very unique cell, hard to miss. And with that white material that you see on the inside of that cell is mucus. Our digestive system secretes mucus. It does it for one of a couple reasons. Reason number one is that mucus, when secreted by our digestive system, it coats the inside of our digestive tract so that acidic foods that we're eating don't burn these cells, which is a protective mechanism. Furthermore, that mucus will mix with the food, therefore diluting it. We dilute our food in our digestive system all the time. And then last but not least, that mucus kind of creates a slip and slide. So it makes it easier for us to move material through our digestive system. On the surface of these cells, you can see a series of what looks like little hairs. These are actually microvilli. These are little finger-like projections that are protruding off of the surface of this cell, allowing each of these individual cells to have a greater surface area so they can do their job more efficiently. And in the digestive system's case, the key job is the absorption of all the minerals that we're trying to reap from our food. So the one thing these cells do inherently well is they absorb the fats, carbohydrates, proteins, vitamins, minerals that are in our food. They also secrete enzymes, and those enzymes are used to further break down food, hence secretion and absorption. Everybody kosher with simple columnar. Because when you're looking at simple columnar under the microscope, what you're ultimately going to see, and the slides that they have in the lab are beautiful slides. They're a beautiful dark purple stain, even darker than what you see here. And what you're going to see is this beautiful pattern of tall cells that have a nice single layer. It's very, very easy to see. The goblet cells are going to be very, very apparent once you look close enough. And once you see it, you really can't unsee it. Juxtapose this to the next one. The next tissue is called pseudostratified columnar. So what does pseudo mean? If I say something is pseudo, if you're a pseudo friend, what are you? You're a fake friend, right? So if I say something is pseudo-stratified, what does that mean?
I mean, it looks like it's stratified, but it's not.
I couldn't have said it better myself. So if it looks like it's stratified, but it's not, then what is it? It's simple, right? This is a very unique tissue. It's called pseudostratified columnar. It is ultimately a single layer of column-shaped cells, but it appears to be stratified. The big thing here, and these cells secrete and absorb as well, this is a layer of tissue where it appears that you're dealing with a multiple layer of cells. But in actuality, what you have is a single layer of cells, and the reason why it appears is because the cells are not uniform in shape. So as we look at this tissue, when you're looking at this tissue as it comes around, this is pseudostratified columnar, and you compare it to this tissue, which is simple columnar. Look at the nuclei. See how they all line up nice and uniform, giving the appearance of a single layer? As opposed to this tissue, where the nuclei are all over the place. Some are closer to the top of the cell, some are closer to the bottom of the cell. So this gives the appearance that you're dealing with a stratified tissue, when in reality what's actually happening here, the nuclei are all over the place, but each of these cells has a different shape. And each of these cells attaches to the basement membrane, making it a simple tissue. What gives this away as columnar, right? And so what you're gonna have to find when you're looking at these tissues is what makes them the same, but then more importantly, what makes them unique. And what tells you that this has to be columnar is two things. What are they? And I've already talked about both of them.
They look long.
Under the tissue, under the microscope, oh, shoot. There we go. Under the microscope, you won't have that benefit of seeing that they're long. So there's something else. There's two things out that are on this.
Oh, the
cilia?
yeah.
Now we're talking. One is you look at the surface of the cell, and you see cilia. The minute you see cilia, you're dealing with columnar tissue. Right? You are dealing with columnar tissue. So with these tissues, you will see cilia. For sure. Once you see that little appendage on the surface of the cell, you know you're dealing with a columnar. Now you just got to figure out which one is it. Is it simple or is it pseudostratified? What's the other thing that you see here that tells you this has to be columnar? The goblet cell. We see a goblet cell right here, right here, right here, right here. Those are all goblet cells. So you have the two key ingredients that tells you it has to be columnar. The difference between these two, though, when you're looking at it under the microscope, this one, simple columnar, is going to have this nice uniform look when you're looking at the nuclei of the cells. This one, you're going to have this nice hodgepodge pattern of nuclei as you go from one cell to the next, due to the fact that each of these cells, yes, they're all taller than they are wide, but they're all uniquely shaped, so the nuclei can end up almost anywhere. This is a tissue that's also very good at secretion and absorption, but where you find it, the best example, hands down the best example of where you find this is the trachea. In fact, this picture here is a picture of the trachea, of the inner lining of the trachea. Think of the trachea as a tube that runs from the base of your throat down into your lungs. Here's the opening of the trachea where air is moving in and out of the lungs. This is the connective tissue that ultimately surrounds the trachea. This is the inner lining of that trachea with these little hair-like cilia. And remember, I was telling you the cilia in the trachea, as we breathe in different things that we don't want in the lungs, those cilia kind of move all those particles up towards the throat so we can clear them out of the throat. These are the same hair-like structures that I was saying smokers destroy, because as they smoke and that tar from cigarettes kind of oozes down their trachea, it destroys those cilia, therefore preventing them from being able to move particles out of the lungs, and that's why they have to hack up a lung all the time. Is everybody good with this particular cell as well? Or this tissue as well? Yes, ma'am.
Can you spend more time why it would be stratified?
Sure. I'm going to draw it on this other picture, on this other side. when you think about simple columnar are a series of cells that are very uniform, taller than they are wide. You had no problem seeing that on the last picture, correct? Pseudostratified, on the other hand, here's that basement membrane. All of these cells are longer than they are wide, but because they're all different shapes, the nuclei are all over the place. So it's a single layer of cells, simple. They're all longer than they are wide, columnar. But they appear, because of the nuclei being all over the place as opposed to being uniform and nice and in a line, it appears that this tissue is stratified when you look at it under the microscope. So we call it pseudo-stratified, false stratified, when in reality it's just a simple tissue. Does that make more sense?
Because they're long.
Because they're long.
So
Columnar.
it looks like it's stacked, but it's really one layer.
Amen.
Okay.
It looks like it's a bunch of cells stacked on top of one another, but in reality, what you're dealing with here is a single layer of cells. It is a simple layer, but we call it pseudostratified because of its appearance. And again, what you're going to have to be able to do under the microscope is be able to sniff out what each of these cells are. And when it comes to columnar cells, there are two things that are going to be dead giveaway clues that you are dealing with columnar cells, one of the two columnar cells. And what are those two ingredients that tell you you're dealing with columnar? Cilia or microvilli and goblet cells. The minute you see an appendage on the top of the cell, the surface of the cell, it has to be columnar. The minute you see a goblet cell, it has to be columnar, has to be. There are no other options. There's no other tissue I'm going to show you where goblet cells are involved. There's no other tissue that I'm going to show you where these hair-like structures or these little finger-like projections exist. So if you see them, it has to be columnar. Your next question to yourself is it simple columnar or pseudostratified columnar? Simple columnar, the nuclei will line up beautifully in a row, giving it a nice uniform appearance, simple columnar. Pseudostratified columnar, the nuclei are going to be all over the place. Once you get that down, these two tissues are duck soup. Any question on this one before we move on? Good. Oops, where did I put that? So now we're talking about the last two epithelial tissues are stratified tissues. These are tissues where you're actually dealing with multiple layers of cells. By and large, the function of stratified epithelium is protection. So for each of these next two, protection is their key function. They don't really absorb, they don't absorb. They don't secrete, but they really do a good job of protecting. Think about it this way. If you have a priceless vase that you need to move from one house to the next, what are you going to wrap it in?
Bubble wrap.
Bubble wrap. And the more layers of bubble wrap you have, the more protected it is, correct? These are cells. The more layers of cells we have, the better cushion our body has with whatever tissue these stratified tissues are protecting, one of which is going to be our skin, the outer layer of our skin. The other one is going to be the inner lining of our bladder. The first one is called stratified squamous epithelium. EPITH is epithelium. So, as this name implies, what is this tissue going to look like if you had to picture it in your head? Stratified squamous epithelium.
It's gonna look flat.
Multiple layers of what shaped cells? Flat cells. In reality, when you're looking at this tissue, the bottommost layer is actually going to be cuboidal. So the bottommost layer of this tissue is actually going to be more of a cuboidal tissue. But then as we stack these cells on top of one another, they steadily flatten out. Until when we get to the surface, the cells at the surface of this tissue are going to be nice and flat. So when we take a look at this tissue, we have multiple layers of cells. You can actually see the individual cells, can you not? At the very, very bottom, the basement membrane, which runs here, each of these cells has more of a cuboidal shape. As you're taking a look at the tissue here on this diagram, the bottom layer are actually very robust cells, but as these cells move further and further towards the surface of the tissue, they flatten out. Hence the name stratified squamous epithelium. Why do you think these cells flatten out as they get further and further away from the basement membrane? They die. The cells on the surface of this tissue are dead. And we want them to be dead. That's actually a good thing. We're actually very, very happy they're dead. Otherwise, this would hurt. Because every time I do this, what's happening to the cells on the surface of my skin? I'm just sloughing them off. Right? If we had a black light up in here, and I was doing this, you would see cells just flying all over the place. The cells just being scraped off of the outer surface of my skin. But they're dead cells, so it doesn't really matter. And the cells on the basement membrane are constantly making new ones. They're constantly undergoing mitosis, therefore continually pushing new cells up into these outer layers. These multiple layers of tightly packed cells work as a great barrier, because anything that tries to penetrate this layer of cells can't really do so, because there's so many layers of cells here, which makes this tissue a phenomenal barrier, protected barrier, which is why we have this primarily where?
On the skin.
This is our skin. This is what the outer layer of our skin looks like. So the best place to find this is the skin. In fact, when we get to chapter 4, one of the things we're going to do is we're going to name all the layers of this layer of our skin. This is our epithelium, which is the outer layer of our skin. It's actually made up of 5 different layers of cells. Stratum basal, and spinosum, and granulosum, and corneum. But this is what the outer portion of our skin looks like. And again, the key function of stratified squamous epithelium is it protects us. There are other places where we find this. As the picture implies, we find this on the inside of the mouth, the inside of the nose, the inside of our pharynx, the inside of our esophagus, all for a protective mechanism. Everybody kosher with this one. Now, when you look in the book, they're going to separate stratified squamous epithelium into keratinized and non-keratinized. I don't want you to stress out about that. They all look the same. The difference between the two is one produces a protein called keratin, which is what our skin does. The other types do not. At the end of the day, we'll get to that concept when we get to the skin, hair, and nails chapter, which will be Thursday. The last one, I'll come back to this one. The last of all the epithelial tissues is called transitional epithelium. Transitional epithelium is also a type of epithelium that is stratified. This is multiple layers of cells. The difference is each of these cells remains robust in that they all maintain their shape. These cells don't flatten out like we saw in the skin. But they don't have a specific shape, all right? Sometimes the cells look cuboidal, sometimes they look squamous, sometimes they look columnar. And what's great about these cells, which makes these tissues very unique, is the cells can change their shape. And they do so depending on the needs of the organ. And the best example of where we find this, hands down the best example, is our bladder. Think about our bladder. What's the function of a bladder? Why do we have bladders?
For storage.
We store urine. So when your bladder is empty, one would assume that that bladder maintains a very round, ball-like structure, right? But as it fills with urine, what happens to our bladder? It has to stretch, right? It has to be able to expand and change its shape. One of the reasons why it's able to do so is the cells that line the inside of our bladder, which are transitional epithelium, they can change their shape, allowing that bladder to expand. Does that make sense? So these cells have the ability of stretching, for lack of better terms. So if I ever ask you about what cell allows for a stretching to occur within an organ, transitional epithelium is where you're going to see it. So here is transitional epithelium. From here out, we can see multiple layers of cells. But when you look at the outer surface of these cells, they still remain their robust shape, as opposed to stratified squamous epithelium, where the outermost cells are nice and flat, are they not? When we get here, nice and robust, just like we see here. But each of these cells has its own unique shape. There's nothing uniform about this tissue. And again, the single best example of where we find it, it's not the only example, but the single best example of where we find this is going to be our bladder. Because these cells, when they change their shape, the bladder has the ability of changing its shape to accommodate all the urine that is filling up. Now, once these cells lose their ability to change their shape anymore, what do you get the urge to do? You get the urge to pee, right? The next thing you know, you pee, the bladder goes back to its original shape, and the process just happens over and over again all day long. These are our 6 epithelial tissues. Before we move on to connective tissue, do we have any questions on these cells? Or these tissues, I should say.
Wait, so the main function of that one is to allow for.
Don't get me wrong, it's a protective tissue. It protects the inside of the bladder too.
Mm-hmm.
But what makes this tissue unique, which is where the questions will really come from, is these cells have the ability of changing their shape. So as the bladder needs to expand, these cells can change their shape. And in doing so, allow the bladder to stretch, to accommodate more urine as the bladder fills up with urine.
Okay.
Mm-hmm. Which is definitely unique to the other, compared to all the other tissues. This is a very unique characteristic of this particular tissue. Now, the one thing I oftentimes gets asked is, do we have things like stratified cuboidal or stratified columnar? And the answer is yes. These two tissues do exist, but they're exceedingly rare. And they usually only exist in areas where one tissue is transitioning into another tissue. Having said that, if you put stratified columnar or stratified cuboidal on a test of mine, it's a guarantee that you just got the wrong answer. I will never show, but here's the thing, you laugh. 20% of you are going to do it a week from Thursday. 20% of you a week from Thursday are going to put stratified columnar or stratified cuboidal on one of my tests, and I'm going to say I told them that that's never the right answer, and I'm going to mark it wrong. It's a guarantee. These do exist. All right, they're exceedingly rare. They're usually found either in large ducts or they're found in areas where we transition from one tissue to another. So they happen in a very microscopic portion of the body, and therefore they're not really necessary to talk about. But I get the question all the time, so my feeling was let's put a slide on it, and then just make sure that you never, ever, ever put that as an answer to my questions. And what I love to do this on the test, is I'll put one of these as a detractor answer, just to see how many of you pick it. And you'll be surprised, about 20% of every class, every test does. Maybe you guys will be the first ones not to. Because every class I tell them, don't do that, and then you do it anyway. Any questions on epithelium? Good. The next category of tissue is our connective tissues. Connective tissues have a very, very unique look. Each connective tissue has its own very, very distinct look. And with it, has its own very distinct set of functions. When you're thinking about connective tissues, we're talking about things now like fat, we're talking about bone, we're talking about ligaments and tendons. We're talking about tissues that make up some of the matrices of our organs. We're talking about blood. And when you think about the difference between blood, bone, and fat, you're talking about three very, very different structures, are we not? So each of these tissues, though, has a few things in common, which is what makes them all connective tissues. First and foremost, they are pretty much uniform with regards to their function. They protect, they support, and they help to bind things together. We help to connect parts of the body to one another utilizing connective tissues, hence the name. We use it as packing material. We use it to transport things through the body. We use it to support and protect the body. And when we think about connective tissue, all of our connective tissues have a couple of things in common. One, all of them have something called an extracellular matrix, meaning we have a series of cells that are suspended within a gelatinous matrix. And that gelatinous matrix is going to be made up of two very important things. One is something called the ground substance. Ground substance is basically a watery, kind of a gooey type of material that suspends each of these cells. The other thing is that each of these extracellular matrices has a fiber, and these fibers bring with it a very unique function to these tissues. Of the fibers, the first one is collagen. And everybody here has heard of collagen. And when we think of collagen, ladies, this one is more for you, because men don't really give two hoots about collagen. The better your collagen is, the better your what is?
Skin.
Your skin. When you think of collagen, you think of skin, do you not?
Mm-hmm.
And if your collagen breaks down, what's the big thing that tends to happen because of collagen breaking down? You get what? You get wrinkles, right? It creates wrinkles. Ultimately, and that, don't get me wrong, collagen plays a big role in helping to maintain our skin and making our skin nice and smooth because it's a primary component of a portion of our skin. But what collagen ultimately does is it brings each of these connective tissues strength. So collagen fibers are utilized in matrices of connective tissues that we need to maintain strength. They give us the ability of having a tough connective tissue that can withstand stresses. The second type of fiber is called an elastic fiber. And elastic fibers, when you think of elasticity, you're thinking of the ability of that tissue to do what? To stretch, right? This gives the ability of these connective tissues to stretch. What's a good example of a connective tissue that all of us can easily touch? That is a good example of a connective tissue that has a lot of flexibility.
Ear.
Our ears, right? Nothing more than, this is a connective tissue that makes up our ears, which gives our ears the ability to stretch, right? Can we not move our ears in all different directions, and then when we let go, technically it goes back to where it belongs, right? Or the tip of our nose, right? Because that's a connective tissue. It's primarily made up of a connective tissue that gives us the ability to move our nose. And we have a whole bunch of different connective tissues that require, we need that tissue to be able to have some pliability. The last one is called reticular fibers. And reticular fibers give us the ability to mold that tissue. We can actually take connective tissues that are primarily made up of reticular fibers, and we can actually mold them. Think of them as Play-Doh. I can take a thing of Play-Doh and I can turn it into various structures, can I not? They got a little bit of elasticity, they got a little bit of strength, but by and large I can mold them to look like I want to look. And we're going to see one particular tissue That's actually called reticular connective tissue. It's primarily made up of reticular fibers, and what we ultimately do is we make this organ by molding reticular fibers and reticular tissue into the shape of that organ. So far, so good. The other thing that ultimately is made up of all these connective tissues is they all have to have a cell. And so each of these cells, each of these different types of connective tissues has a very unique cell. The cell of a bone is called an osteocyte. The cell of a cartilage cell is a chondrocyte. The cells in our blood are red blood cells, white blood cells, and platelets. So each of these tissues has their own unique cell, and those unique cells, A, make that tissue unique, but B is what allows for that tissue to function and grow and expand or regenerate, whatever it might need to do. So, we're going to take these one at a time. Our first tissue is bone. If you miss bone on the lab exam, shame on you. Ray Charles could see bone. For those of you who don't know who Ray Charles was, he was a blind piano player. But Ray Charles could look at bone tissue under a microscope and say, oh, that's bone. It's a very, very unique tissue. One that you should not ever miss. Bone and blood should never be missed. And they ask one or the other, or both of them, on every lab exam. Bone is composed of osteocytes. All right, so that's the name of the cell of bone. Is an osteo always means bone, cyte always means cell. So osteocyte literally translates to bone cell. And these bone cells are found within a extracellular matrix that's very, very hard because it has a lot of calcium and salts in it. And it has a huge number of collagen fibers, as I just described. Collagen fibers give connective tissues what?
Wait, say that again?
There's a large amount of collagen fibers within our bone tissue because collagen fibers give connective
Strength.
tissues what?
Strength.
Strength. And when you think of bones, the one thing you should always think about is that they are strong structures, correct? Thank God our bones are strong and not soft. And again, bones, if you don't know where bones are located, I can't help you. And if you don't understand yet that bones protect us, I can't help you with that either. When you're looking at a bone tissue under the microscope, what you're ultimately looking at least in my screwed up head, this looks like a series of tree stumps. If you think about a tree that just got chopped down, where you have the center of the tree stump surrounded by the different layers of that tree stump. And each of these layers, the more layers you have, the thicker the bone is, the stronger the bone is. And as we look inside each of these little black dots that we see, these are all the individual bone cells or osteocytes that exist within the layer of this bone. Each of these little cross sections that we're seeing are known as an osteon. And we'll talk about this in greater detail when we get to the bone chapter. We'll actually look at this more microscopically, and we'll actually peel off all the different layers and show what's going on there and why it's going on. For now, what I need you to be able to do is recognize bone tissue under the microscope. But everybody can see what I'm talking about here, right? Bone's one of the easy ones. And again, they ask it routinely on the lab exam. The next one is cartilage. There's 3 types of cartilage. And each of these 3 types of cartilage has a very unique characteristic to it. And with its unique characteristic comes with it the reason why we find it in its unique places. So when we think about cartilage, the first one is called hyaline cartilage. So these are going to be always going to have chondrocytes. Hyaline cartilage is primarily made up of, collagen fibers, meaning hyaline cartilage has a great amount of what? Strength. Hyaline cartilage is going to be found in areas where the cartilage that we're using needs to be able to withstand a lot of stress. So, some good examples. This is gonna make up our joints. When we think about the bones that make up a joint, take our elbow, right? We have two bones that make up our elbow. Covering the surface of each of those two bones is cartilage. That's the cartilage, that's the cartilage that gets roughed up when we have arthritis. The cartilage at the end of our bones, of all of our joints, is made up of hyaline cartilage. Another good place where we have hyaline cartilage is going to be on our rib cage. Our ribs attach to our sternum utilizing cartilage. We have cartilage all in the center of our chest, right underneath the skin of our chests. That cartilage connects the ribs, and it has to work as an extension of the rib. We can't have this be all loosey-goosey because this rib cage is protecting our heart and lungs. This is hyaline cartilage, very strong cartilage. Another good example of where we find hyaline cartilage is in babies. When babies are born, right? Baby comes right out the chute, day one. Is that baby made up primarily of bone or primarily of cartilage? Cartilage. They're primarily made up of cartilage. Where their skeletal system would be is really, it looks a lot like cartilage. Well, it is cartilage. And at the end of the day, that's why babies can't stand, right? Fresh out the gate, they can't stand. But then as their bones grow during the first year of their life, they gain the ability to be able to withstand the pressures from standing. Yes, ma'am.
So do babies do or do not have more bones than adults have?
They don't have more bones. The problem is each of their bones looks like multiple bones. So when you take a look at your femur, how old are you?
20.
20, so you're done grown. So here's your femur. With me? It's one bone, which makes up the thigh, your thigh bone. But when you were 6 months old, your same femur looked like this, if you took an x-ray. It looked like that. If you took an x-ray of your femur when you were 6 months old, it looks something like so. So it looks like you have 1, 2, 3, 4, 5, 6 different bones. But what you don't see on the x-ray is that connecting each of these bones to one another is cartilage.
Look at that.
So you have one giant bone, but it's just in multiple pieces connected to each other by cartilage. And these cartilaginous places are known as what? Well, they're made up of hyaline cartilage, but what are they called? You've heard of the term. They're called growth plates. You've heard of growth plates, right?
No.
Well, growth plates are parts of bones in non-adult human beings where these bones are now able to grow. The reason why you got taller, because when you were born you were this tall and now you're however tall you currently are, this cartilage gets turned into bone, making this bone longer. And then when you're done growing, these growth plates go away. And now what you have is just one solid piece of bone. So you didn't have more bones, but each of your bones weren't completely formed yet. And it takes, for ladies, you're done growing at give or take 18. For the gentlemen in the house, 20, 21, 22 at the max, right? We're done growing in our late teens, early 20s. At that point, you're referred to as skeletally mature. Well, this cartilage that separates each of these bony parts from one another are made up of hyaline cartilage. It's a strong cartilage that maintains that bone in its given shape. Eventually, when we get to the bone chapter, I'm going to show you my son's hand. My son fractured his hand, and I'm going to show you where his fracture is. And you'll see that each of the bones of his hands, instead of it looking like one bone, it looks like 3. Each of the bones of his finger, so it looks like he has 3, 6, 9 bones instead of 1, 2, 3 bones. And it's because when he fractured his hand, he was 11, right? And so you'll see the growth plates when we get there. So when we're looking at hyaline cartilage, what we're seeing here is a matrix here in this bluish purplish color. This is that extracellular matrix, which is primarily made up of collagen fibers, giving this cartilage a lot of strength. Intermixed and suspended within that matrix are a series of cells. These are individual chondrocytes. which allow for us to add to this matrix as we need to. If we need to make the cartilage stronger or repair it, what have you, these chondrocytes will do that for us. The example your book uses is the cartilage that connects your ribs to the sternum, which we have even into adulthood. Again, some other good examples, the joints of our body, the coverings of the bones are at the joint is hyaline cartilage, and babies are primarily made up of hyaline cartilage. That's what's connecting all their bones to one another. Everybody cool with hyaline? The next cartilage is called elastic cartilage. An elastic cartilage is primarily made up of elastic fibers. Giving this type of cartilage the ability to do what? To stretch. It gives this cartilage the ability to stretch. There's two very good places where we find it. The first one is our ear, and the second one is something called our epiglottis. Everybody can see that their ear has the ability of changing its shape and being elastic, correct? That's because the cartilage that makes up our ear is primarily elastic cartilage. It is elastic cartilage. Anybody know what our epiglottis is? The epiglottis is a little flap that sits at the top of our trachea and esophagus. That flap opens and closes depending on if we're swallowing something or if we're breathing in and out air, and allows us to dictate whether or not stuff moves into our trachea or our esophagus. And it is primarily made up of elastic cartilage as well. To see elastic cartilage, it looks exactly like what we see here. The difference is there's going to be a lot of black lines rolling through it. And these black lines are little elastic fibers, which give this cartilage the ability to stretch. And the last of the three is called fibrocartilage, is what we see here. So a third of the cartilages is fibrocartilage. Fibrocartilage is also primarily made up of collagen fibers, which means this cartilage has a lot of very, very good strength to it. But this is cartilage that we ultimately create puck-like portions with. There are two places in the body where we take cartilage, we compress it, and we turn it into what looks like a hockey puck. The two places, one of which the board is showing you, is the spine, specifically the discs of the spine. And the other part is something called our pubic symphysis. What's our pubic symphysis?
I know what the pubic is, but.
Well, what's your pubic? It's this neck of the woods, right? What does symphysis mean? By symphysis means we're bringing something together. So what do you think we're bringing together with our pubic symphysis?
Pubic bone?
The pubic bones, right? Our pelvic bones, we have a right one and a left one, and they're connected to one another. And in the front, right down in this neck of the woods, there's a puck of cartilage that connects the two bones to one another. And it's known as our pubic symphysis. But in areas of the body where we have little pucks of cartilage that connect two bones to one another, like we do in the spine and we do in our pelvis, this is the cartilage that we're using. What we're looking at is cartilage that's been heavily compressed. Here's all the little chondrocytes floating in the middle. And what we're seeing here is primarily collagen fibers in its extracellular matrix to give it a lot of strength. And we'll see all three of these cartilages in lab today for sure. Everybody kosher with cartilages? Good. When it comes to the dense connective tissues, there's only one you're responsible for. They long ago got rid of tendons and ligaments. This is what a tendon and a ligament looks like, but they got rid of it. The one you are definitely, but the one that the connective tissue that we're definitely going to be responsible for is our loose connective tissue. So we do need to know about these. And the three varieties are areolar, adipose, and reticular. And these are the last, though, there's four more, one after this. When we think of adipose, what are we talking about?
Fat.
Adipose is fat. Reticular connective tissue is going to be connective tissue that is primarily made up of reticular fibers. We'll get to that in a second. Areolar is the next one. This is areolar connective tissue. It's a very, very unique tissue. Areolar connective tissue this has a mix of collagen and elastic fibers, a nice mix. So we take a look at this, we can see the collagen fibers here with these dark purple strands, and then these little black lines that we see going through, these are elastic fibers. So this is a tissue that has a mixture of a lot of strength, but a lot of flexibility as well. The best place where we find areolar connective tissue is something called the basement membrane. And the basement membrane is also known as the lamina propria. Both are good names. So as we look at all of our epithelial tissues, so for example, here's our epithelial in red. That epithelial tissue, all of our epithelial tissues have to connect to a basement membrane. That basement membrane is made up of areolar connective tissue. Very unique look. When you see this under the microscope, it's going to have this kind of cotton candy-like appearance. Again, this is another one they love to pick, and they're showing you the same basic premise that I tried to draw here, up in here. Here we're seeing the skin, and the skin, the epithelial tissue, has to connect to the basement membrane. This basement membrane is made up of this tissue. So any place where we're trying to connect epithelial tissue to deeper tissues, we do so using areolar connective tissue. Adipose, this is another one. Oh, sorry.
I'm sorry, does that say lamina?
Lamina. Next word is propria.
Okay.
Yes, ma'am. Can I erase this? You good?
Yeah.
Does this have anything to do with the areola?
No. No.
Okay.
No, when you're thinking about the nipple, now you're talking about, now you're talking about epithelium.
Okay.
Yeah.
Oh,
Why it got that particular name, I don't know. I wasn't at the meeting. I would have named it something different, I'm sure, but. The next one is adipose. If you don't know where fat is found, I can't help you, all right? Fat is found in a whole host of different places, all right? If they ask you, where do you find fat? You can say your abdomen, surrounding your organs. There's a whole host of places where we find fat. All right? Fat in general protects us. It gives us a layer of cushion either around our organs or underneath our skin. It helps to cushion or create like a little bit of a cushion to the skin. We use it as energy if necessary. The single easiest place, if they do ask you where do you find fat tissue or adipose tissue, is breast tissue, since the breasts are primarily made up of fat. Insulation, all kinds of things. Fat is a very easy one to see. When you're looking at fat, what you're looking at here are little droplets of fat represented in white. Surrounding it is connective tissue, and here are the little fat cells that exist surrounding these little droplets of fat. Again, fat's one that should pretty much go without saying. You should have come into class knowing where you find fat and having some ideas as to what FACT does for us. It's just a matter of recognizing this under the microscope. I'll never ask about this one on the lecture exam because it's too easy. I will definitely pick harder tissues. But on the lab exam, they ask this one pretty regularly. And it's surprising some of the answers you all come up with. Again, this is another one that Ray Charles could see. The last of our connective tissues here is referred to as reticular connective tissue. And as its name implies, it's primarily made up of reticular fibers. And one thing we said that's great about reticular fibers is we can take reticular fibers and just like Play-Doh, I can mold them into shapes and structures I want them to look like. The best example of where we find this is going to be in two areas. One is our spleen, and one are our lymph nodes. These are the two most abundant areas where we find this tissue. Reticular tissue has a little bit of elasticity to it, so these tissues, these organs are flexible. They also have a little bit of strength to them, so these organs can hold up on their own. And when you look at reticular connective tissue, we're seeing the spleen here. The whole spleen is made up of this reticular connective tissue, this tissue that we've just kind of molded into this spongy-like organ. Lymph nodes look very similar, which are very hollow on the interior, which is represented by all this white empty space. We see collagen and reticular fibers throughout. And each of these little cells that we see are the cells of our immune system floating within our spleen, or if this was a lymph node, our lymph nodes. So the cells of the tissue are ultimately going to be the tissues of our immune system, or the cells of our immune system. The tissue is primarily made up of reticular fiber, which is a combination of elastic and collagen fibers to give it strength and elasticity. These generally are hollow structures, allowing these cells to freely move within this organ. The one that we're looking at under the microscope today, it's got a very orangey appearance as opposed to this purplish appearance, but the actual look of it's very similar otherwise. And then the last one before we go to lab is blood. Blood you should not miss. Do not miss blood on the test. They ask it regularly. Blood smears look like what we see here on the right. If you don't know where we find blood, heaven help you. All right? Please do not be my family's nurse if you don't know where we find blood. But ultimately, blood has the same characteristics. It has an extracellular matrix, which we refer to as plasma. This is the fluid that these cells are floating within. There's no real reticular connective, those fibers in blood. It's unique in that regard. The cells are a combination of three. We have red blood cells, which carry oxygen and carbon dioxide. We have white blood cells, which are immune in function. And we have platelets, which are little fragments of cells, which we use to clot our blood should we damage a blood vessel. So these are our connective tissues. What we have left is 4 more tissues. We have 3 muscles and a nerve, which we'll get to on Thursday. I'm not going to worry about those today, and you shouldn't worry about them either today. What we are going to look at in lab today, we're going to take a quiz. And after the quiz, what we're going to start doing is looking at each of these epithelial tissues. We're going to look at each of these connective tissues under the microscope and start getting used to those. We have 1 week. We have 1 week of exposure, or another way of saying is 2 lab periods, or another way of saying it is 3 hours to be able to see these tissues under a microscope. Because once we get past the tissues,